Good thinking. Good planning ahead. All right, so morning.
Morning.
I'm going to start recording. It'll let me. It says it's starting. Anyway, once it actually starts recording, I'm going to repeat your question. So that's, available to anybody who watches this video afterwards. There we go. Now we're recording. So, Vance, like you said, next week's lab, I'm not sure off the top of my head if it has a pre-lab or not. But if it does, I'll make sure to post that lab. Well, I'm going to post it soon anyway, but definitely if it has a pre-lab, I'm going to make sure to prioritize that so you guys have time. We have less than a minute, so I'll go ahead with the other normal announcements. Attendance is going to be the way it usually is. If you're online, send me the words. Make sure you send it before 9 a.m. My laptop's not working, so White, I know you're here, but if you could, just send me an email to remind me if you can, but if not, whatever, it's up to me, so I'll figure it out. And right now, you're the only person here, so it should be pretty easy to remember. What other announcements while we're recording? There's not much to say, really. If you weren't at lab on Monday, online that is, then you need to be online for lab today at 2 p.m. unless you have done the lab and I've graded it and told you don't need to be there. So my suggestion to you, if you haven't done the lab yet, I would just go ahead and knock it out. That way you don't have to show up at 2 p.m. online. But I don't know. That's it for lab. That's it, really. We're still stuck in chapter 9, which seems to be the chapter that never ends. The next exam is going to be on chapters 8, 9, and 10. So obviously we got a little bit before we get to that. What else? Independent work is no longer mandatory, but it is extra credit. And some of you really need extra credit, yet are not doing it. And I want to remind you now, again, like I have throughout the semester since the beginning, don't cry to me about your grade at the end if you haven't been trying to fix your grade since the beginning. Whew, got that out. Any questions before we officially get started? All right, nice and easy. Can someone, not yet, but there we go. Can someone please confirm that you can see the PowerPoint slides?
Yes.
Thank you, Mr. Vance. All right, let's jump into it quickly. I want to remind you what we're talking about here, though. This main bullet point, the variations on Mendel's themes. To remind you, this first bullet point, when we talked about genetics and heredity, you got the really basic version of stuff where we talked about this one gene controls this one character, and this one character only comes in 2 varieties, right? Like the pea plant either has a purple flower or a white flower, right? Or you either have a chin dimple or you don't. Those are the basic things we talked about. And then once you got a good understanding of that, theoretically, then we moved on to the variations, which was saying, hey, look, most things are not that simple. Usually there's other things going on. And that's where we ended up with this when we talked about some of them. And as I briefly remind you what we said, I also want to briefly remind you how the questions are going to go on the exam for this section. So basically what I'm going to say is this thing mated with that thing, and these were the results. And based off of that, you're going to have these choices. Your choices in the multiple choice exam are going to be incomplete dominance, complete dominance, pleiotropy, codominance, polygenetic inheritance, or epigenetics, right? And you need to be able to figure that out based off the information I give you. For example, incomplete dominance. Well, no, let me start with the beginning. For example, the first thing we talked about with this main bullet point right here, genetics and heredity. If I mix a white flower and a purple flower, and 100% of the offspring are all white, right? Then you might, or excuse me, if I mix a white flower and a red flower, and 100% of the offspring are white, then I would say that is an example of complete dominance, and that's how you would answer it on your exam. Then if I said, all right, I mixed a blue alien with a yellow alien, right? Some of the aliens were blue, some of the offspring were blue, some of the offsprings were yellow, some of the offsprings were green. I'm like, all right, well, green is a mixture of blue and yellow. That is an in-between of the 2 phenotypes. Therefore, that is an example of incomplete dominance. Then I could say I mixed 2 other aliens. One was blue, one was red. Some of their offspring were blue, some of their offspring were red, but some of them were blue with red spots. I'm like, oh, okay, that must be codominance, which isn't listed right there, but the blood groups, that's an example of codominance. So if it shows both phenotypes, that's codominance. Now, pleiotropy, it's hard for me to give you a good example, so I just give you the book example, because otherwise it'll take up too much time. The book example of pleiotropy was sickle cell syndrome, which was there's one gene, but instead of that one gene controlling one thing like eye color or chin dimple or freckles or anything like that, that one gene controlled a lot of different traits. And again, it was sickle cell. So that one gene caused the sickle cell shape of the cell, but then you had this other long list of symptoms for it. And that's where we left off. So again, really quickly, with pleiotropy, I'm going to give you some off-the-wall thing that probably doesn't even exist, but you're going to be able to recognize it because I'm going to say there was this one gene, and then because of this one gene, there's all these different things. That's it. All right. Are there any questions now that we're caught up? Does anybody have any questions about all the stuff we just talked about, or that we've talked about previously? All right, let's jump into polygenetic. Actually, that is a misprint. I need to fix that. Polygenic inheritance, not genetic. Though polygenetic does make more sense. Polygenic inheritance is what it's supposed to be called. And it is sort of the opposite of pleiotropy. And maybe that's how the question will be worded. I don't know yet. But remember, pleiotropy, one gene, many characters. The opposite of that is polygenic inheritance. Polygenic inheritance is basically more than one gene on one character. Or as your book puts it, the additive effects of two or more genes on a single phenotype. Again, like I said, that is the opposite of pleiotropy. Pleiotropy is like we got one gene that affects all these different phenotypes. One of the things, I think this is the way the question is worded on the exam, you'll be able to recognize something that has polygenic inheritance because if you graph it and like graph a population, there'll be a bell curve. What do I mean by bell curve? This is where the word curve comes from for the people who curve grades. You know, that's the number. That's if that's the graph, a curve, right? Bell curve. And I forget exactly how it's worded on the study guide, but I highly recommend doing the study guide as always, so you can see how it's worded. But yeah, when you're looking at polygenic inheritance, it's a bell curve. So for example, let me do the opposite of that. If we were talking about plotting the number of flowers in complete dominance, it's like, well, some of them were purple, it'd be a bar graph, right? And some of them were white. Pretty simple. This is gonna be a pain, all right? Or if it was, I don't know, incomplete dominance, then you might be like, all right, well, some of them were white, some of them were pink, and some of them were red, right? So there's only 3 bars, right? Now here, what we're getting at here, when there's a curve like that, what you really are seeing is just a bunch, just so many bars. Imagine like there's so many bars that you actually have to draw a line over the top to show the curve, right? Imagine these are all bars that I have written over here to the right, right? Imagine, again, this is 2 bars, this is 3 bars, this is just, I don't know, there's a lot of bars, right? That's the point, right? So there's so many phenotypes, so to speak, that there's not 2 phenotypes where you could graph it like that, or 3 phenotypes where you could graph it like that. There's a bunch. And here's a good example, height. And I keep using this example throughout chapter 9. Human height, right? It's not like all humans are tall or short, meaning they're all either 5'5" or 6'5", right? It's not like that. Those are not the 2 height phenotypes for humans. It's not even 3, right? It's not like everyone's either 5'5" or 6 foot or 6'5", right? Even that, we have way more than 3 phenotypes. If you were to graph the human population and like how many people, I don't know what the average height is, maybe it's 5'6", I think. If you were to graph everybody, you would probably find that most people are the average, right? Most people are 5'6", and then very few people are, I don't know, 6'10", and even fewer people are 4'5", whatever it may be. That's what I mean by a bell curve. And that's why it's a bell curve, because there's not just 1 phenotype or 2 phenotypes or 3 phenotypes or even 4. There's hundreds. So that being said, let's look at a better example. This is exactly what I was saying. But even in this situation, your book is still simplifying it. So we're still looking at height. And this, again, this is just an example, a hypothetical example, because there's more than just 3 genes controlling height. But hypothetically, let's talk about that. Let's assume there was 3 genes controlling height. And that's more than we've seen yet, right? So at the beginning of the chapter, we kept talking about 1 gene, 1 character, right? Then we at least upped it a little bit and talked about 2 genes and 2 characters. So we had that Punnett square that was really big that had 16 squares, right? Now we're upping it even again, one more time. Now we're talking about 3 genes. Again, still 1 character. So even with 3 genes, which again, height is much more complicated this, but if we were to simplify it and say that there was only 3 genes and then do a Punnett square for each of those, these are all the different outcomes just with those 3 genes, right? This is how it would look. And if you were to graph that, again, this is what we mean by a bell curve. You would find that most adults are at that medium, right? At that average height. And then fewer of them are a little bit taller and a little bit shorter. And even fewer of them are even taller or even shorter. And way fewer of them are even taller or even shorter. That's all that means. And again, keep in mind what you're seeing there is a very simplified version. There's many more genes involved in height. And there's also a lot more than genes involved in height. There's also things like, you could have diseases that may have stunted your growth. You could have a virus that could have stunted your growth. Or more commonly, nutrition. Nutrition has a lot to do with your height. But height is not important for this exam or for this semester. What's important is you understand the concept of polygenic inheritance, which is more than 1 gene controlling 1 character. Another example might be, intelligence, like IQ. I'm sure there's quite a few genes involved in that. Anyway, any questions about that? All right. Let's move forward. Took us a while to get here, but it seems like we're moving at a pretty good pace now. Now we're going to talk about the last of variations on Mendel's themes. And then after that comes the really fun and quick part, is the chromosomal basis of inheritance. And well, you'll see why that's going to be quick when we get there. But let's talk about epigenetics. And it's funny, I'm glad epigenetics comes next, because what I just mentioned earlier has a little bit to do with epigenetics. So like I said, height, that's not all genes, right? That's also environment. Like what's your nutrition? That involves, your nutrition can affect your height. Sorry, that's good. It's good to get some a soundtrack every once in a while. Next, anyway, so yeah, height is a good example of it's not just your genes. There's also other things involved. Same with your intelligence, right? IQ. It's not just your genes. There's other things involved. So let's talk about that. Epigenetics. Here we go. This is from your book. Many characters are the result of a combination of things that are from heredity, like your genes, and the environment, right? So it's not all genetic. Despite the fact that we've been focusing on genetics this whole time, this is basically saying, yes, but keep in mind that it's not always all genetics. This goes back to that old saying, what is it? Not saying, that term. You guys ever heard nature versus nurture? Have you heard that? I don't know. Has anybody online ever heard nature versus nurture? There's a big debate. I think the most common one now is homosexuality, right? I guess it's probably shifted from between that and gender. But anyway, homosexuality, for example, you would say, well, is there genes involved? Is it your genetics that make you homosexual, or is it your environment, nature versus nurture, right? So anyway, again, like your book says, whether or not these things are more influenced by genes or environment or nature versus nurture is a very old and hotly contested issue, like you just said. Your book uses a great example of identical twins. Now, if any of you have just met identical twins or spent a little bit of time with identical twins, then this doesn't mean much to you. But anybody who's spent a lot of time with identical twins, like they're besties or they're in the family or whatever, then you can tell, man. You can see clearly these are identical twins, but you know which one is which. Because even though they have the same genes, those genes have been expressed differently, right? So that's why they are slightly different. And here's something I'm about to tell you that the book doesn't say, and this concept is going to come back later in the next chapter, the way I like to describe genes and DNA in general. Remember, your genes are instructions on how to build proteins, right? And those proteins are usually enzymes that dictate all kinds of things, like your eye color and your chin dimple and how tall you are and how smart you are, how hairy you are, whether you have a bent little finger, all those things that make you you, a lot of it's controlled by genes. Now, what you're going to learn next chapter, you're going to learn a lot about these genes and a lot about chromosomes in general. But you can think of them as a set of instructions. Remember, you have 23 pairs of chromosomes. So you can think of each one of those pairs of chromosomes, each one of those chromosomes is a book. It's a set of instructions. And let's say you need to build an enzyme that breaks down lactose. Where do you find that? Well, you go to book number 5, which is chromosome number 5, and you go to page number 38. And then right there is the instructions on how to build the enzyme that breaks down lactose. Of course, I'm just using that as an example. I don't really know where that gene is. You look at identical twins, they have the same sex, but identical twins, same instructions. However, just because they have the same instructions on how to build this, that, and the other thing, doesn't mean they're always building the same things at the same time, right? So one person might have a gene that says, all right, you need to build this protein, and that protein might be disease causing, right? So they both have that gene, but for whatever reason, one twin might be reading those instructions and building that protein and causing that disease. Meanwhile, the other identical twin who has the same set of instructions, for some reason, for whatever reason, even though they have those instructions, their body's not reading those instructions. So they're not building that protein, so they're not causing that disease. But anyway, I got off on a little bit of a tangent there, but that's going to be very important on the next chapter. And again, for here, like we're saying, when we're talking about epigenetics, actually, that really was a horrible tangent now that I'm thinking about it. When we're talking about epigenetics, again, what we're saying here is it's not just genes that control the phenotypes, it's also the environment. So really, the tangent I just went off of was horrible because I was still discussing nothing but genetics, sort of, because I didn't get into it. But what I mean by that, let me go one more step further to clarify things. When I say that one, they have the same set of genes, but one's being expressed and the other one's not, it's because of the environment, right? It's whatever that one identical twin is doing that the other one is not, whatever it's been exposed to, that's what's causing their body to read that set of instructions, while the other identical twin is not reading that set of instructions. So again, that's going to be more important later. For now, for this chapter, what you need to know about epigenetics is simply that when we talk about something, a phenotype, it's not always just genes, sometimes it's also the environment. So any questions about that? We'll talk a little bit more about it here in a second, but are there any questions so far?
Not really about that, but are you going to do keywords today?
Yeah, thanks. Let's do twins as the first keyword. Sometimes I get rolling and forget it. Thanks. So twins is the first keyword for attendance today.
Thank you.
Thank you. Next slide. In general, only the genetic differences that are inherited in an affected environment are passed to the next generation. So what we mean by that is, I'm trying to think of a basic way to put it for you. Obviously, we already know because we've already talked about this. It's only your genes that get passed down, right? It's only the genes that are inherited, generally speaking, right? Again, if we're having this discussion about nature versus nurture, is it the genes or is it the environment? It's usually only the genes. However, some relatively new information in biology is that we've begun to recognize the importance of something called epigenetic inheritance, which is the transmission of traits by mechanisms not directly involving the DNA sequence. And let me try to explain it this way. And this is not an exact accurate way to describe this, but it works with what you guys have been taught. So I'm going to teach it this way. Try to remember these 4 little proteins. Excuse me, actually, technically there were 8, but I can't really draw. Imagine if this is 3-dimensional. There's like 8 little proteins right here. That is so horrible. All in a cube, right? And those things are called histones. Then you've got this molecule called DNA. And it wraps around. Remember? You guys have seen that picture. Remember? And then it would wrap around again, and there'd be 8 more little balls right there where I just did the double loops. And then there'd be 8 little more balls right there where I did the double loops. Try to imagine that picture, or even go back. I should have put it in here. Hold on. Let me just make sure it's not on the next slide. It's not. Okay. So try to imagine that there's these balls of protein that the DNA wraps around. You already know this because remember from chapter 8, when we talk about prophase, remember we talked about how the DNA unravels, right? It unravels, it tries to leave those proteins alone, or excuse me, it ravels up in prophase, because when it's not in prophase, it needs to be unraveled. Why? Because there's a cellular machine, basically, that clamps onto this DNA, and it reads the code. And that's the thing that makes the protein, right? Long story, you'll learn about that later. But basically, there's a machine that clamps on there and reads the code, and that's how we get the protein. Now, imagine if that DNA is really tightly wound around that protein, then the machine just can't get to it. Like, yes, there's a code there. There's a set of instructions here or here or here that say build whatever protein. But if the machine that reads the code can't get to it, then it doesn't matter. Again, going back to my analogy of your chromosomes being a set of instructions, a book of instructions. You might have instructions on how to build, I don't know, whatever protein on page 77 of book number 2. But if those pages are stuck together for whatever reason, right? Maybe they're glued together. It doesn't matter that you have the set of instructions. You can't open it up to read it, right? That's the same thing with epigenetics. Yeah, you might have the gene for whatever protein, but because of whatever's happened in the environment, you've either loosened this, or you've tightened it, or you've completely gotten rid of it, right? So you've made it either easier or harder to get to that gene. And that's what epigenetics is. That's what we mean. Really, that's the new information about how the environment affects your genes. But anyway, some of the newer information is that we've come to learn whatever it is that causes those things, that DNA to wind tighter or looser around those proteins, sometimes that also can be inherited. So it's not just the genes. We're starting to learn it's not just the genes. So for example, smoking is bad for you, we all know. But even if it doesn't mess up your genes, it could mess this up. And we're learning that that could possibly be inherited. Now, again, to remind you, now that I'm done talking about that, I want to remind you what I just told you about the histones and the nucleosomes and the DNA. That's not 100% accurate, but it's the best way to describe it, because so far the only interaction you guys, or the only information you've had about DNA in this context is that it is wrapped around protein. So that's just the best way to describe it. Also, before I move forward, I'll remind you, for the exam anyway, everything I've just told you has been a big waste of time. It's not, but for the exam, you only have to worry about strictly epigenetics, which is just that we're talking about the influence of genes and the environment. So if you're taking notes, strictly for the exam anyway, you would write epigenetics equals gene and environment, right? And put a big emphasis on the word and, because we're talking about both. So any questions about that? Okay. Here's a more accurate version of what happens as opposed to the DNA histone version, which is a little bit less accurate. I'm going to go through this quickly because you don't need to know it for the exam, but it does come from your book. Chromosomes can be chemically modified by adding or removing chemical groups on the DNA and or protein components. And what do I mean by protein components? Exactly those things I showed you earlier, where the DNA wraps around the proteins. So DNA, all this stuff can be chemically modified. And if you chemically modify it, you adjust the shape. And remember, shape is it's always very important for proteins, but now it comes to learn, that it's also important for DNA too. So yes, these changes can be made. The environment can play a role in making these changes. And again, this is going back to the identical twin thing. This may explain how one identical twin can suffer from a genetically based disease, whereas the other twin doesn't, despite identical genomes. So again, they might both have a gene for a certain disease, but for whatever reason, only one identical twin's machinery, cellular machinery, can read that gene, while the other ones cannot read the gene. So simply, epigenetic changes DNA and chromosomes, while genetics is strictly the sequence of the pairs, the A's, the T's, and C's, and the G's. So when we're talking about genetics, that's strictly the code, right? The A's, the T's, the C's, and the G's. When we talk about epigenetics, it's that plus just the actual physical shape and components of the DNA and the chromosome. So any questions about that? All right, epigenetic modifications and also changes in gene activity that result from those changes can be carried on to the next generation. This is what I was saying earlier. Now it's just in writing for you. So we're learning that, yes, some of this is inheritable. But unlike alterations DNA sequences, chemical changes to chromosomes can be reversed. So that's good news, depending on the situation. So maybe if you've done some damage to your DNA from smoking, excuse me, not to your DNA necessarily, but your chromosomes. If you've done some damage to your chromosomes by smoking, then according to this bullet point, then if you were to stop smoking, now that doesn't mean you should go out. That would be genetic.
We couldn't hear you for about 30 seconds.
What's that?
The sound keeps like going in and out.
Yeah, I couldn't hear you for about 30 seconds. It just came back and you said that would be genetics.
Oh, man, that stinks. Well, here's the good news. For this slide, none of thing is important for the exam. I would go back and repeat myself, but none of this will be on the exam. I'm just kind of wrapping up this section on epigenetics. But thank you for letting me know. So I guess
I'm still interested, though. What would happen if you stopped smoking?
Well, if you stop smoking, if we're talking about epigenetics, then some of the epigenetic damage you've done could be reversed. However, like I was saying, like I was finishing up, don't go. I hope not. All right. This next part is going to be very quick. Basically, this third portion of the chapter, not completely, but a lot of it is just repeating what I've already said, except now it's going to say it in the context of chromosomes. So instead of just saying, this gene is inherited and that allele is inherited, what it's going to say is the reason this gene is or this allele is inherited is because it's on this chromosome, and here's what the chromosomes do. So because of that, I'm going to go through this quickly, because even though your book didn't really talk about chromosomes very often in those first two bullet points, I did. So everything I'm about to say now, for the most part, is going to be a repeat. Are there any questions about that? I'm going to try to pause more often to make sure you guys can hear me. All right. The next word for attendance, I'm not going to say it. I'm going to circle it. That way, I'm going to make sure everybody's paying attention with all their senses, not just listening. But there's your next word for attendance that I just circled. All right. Anyway, moving forward. The chromosome theory of inheritance states this, and you already know this, but again, we're repeating it. It's making it very clear. Genes are located at specific positions on chromosomes. We already know this. I'm going to do a horrible job of drawing the chromosome here. So let's say, I don't know, that's chromosome number 18 for whatever reason. I hate that. So forget those lines. Those lines aren't supposed to be there. That's a mistake. Actually, no. Let me utilize those lines. All right. So let's say that's chromosome number 18. And let's say gene P is found right there at the tip of chromosome number 18. That means for all humans, unless there's something wrong with a particular human, if you were to look at chromosome number 18 at that location, at that loci, as a singular, or locus if it's plural, if you were to look right there for every human, you would find gene P. Meanwhile, if you were to look at every human right here, chromosome number 18 towards the center, you would find chromosome number Q, for example, right? That's what we mean by genes are located at specific positions on chromosomes. You already know this. You also already know, excuse me. Well, first of all, did everybody hear that first bullet point? Or did my sound cut out?
Yeah.
Okay, thank you. Next bullet point. As you already know, the behavior of chromosomes during meiosis, right? When all these chromosomes split up, that's meiosis. And during fertilization, which is where, they come back together, so to speak, that behavior, those things that happen in meiosis and fertilization, this is what accounts for the inheritance patterns. So we already know this. So every time I showed you earlier in this chapter, and even in lab, when I said, all right, this person's homozygous for this person's heterozygous. And when I did the Punnett square, right, I would even show you, I would say, okay, this allele separates from this allele. This allele separates from that allele, because that is what happens during meiosis. And then when you actually show the Punnett square and saying, all right, you got the big T from the dad and the little t from the mom, that would be fertilization, right? Those little squares, I don't know why I'm trying to describe it so you can picture it in your brain. I'll just draw it, right? When we're talking about Punnett squares, I'm not going to draw the whole thing, but I don't know if there was a big T, little t right there. That represents a fertilization, right? Because the big T may have been the sperm from the mom, or the sperm from the dad, and the little t may have been egg from the mom, right? So that's fertilization. So you're already familiar with this. Again, this is why hopefully I can go through it quickly. It's also, well, first, did everybody hear the second bullet point?
Yes.
Yes.
Thank you. Finally, it is the chromosomes that undergo segregation and independent assortment. And remember, segregation is just the thing that says, all right, I have blue eyes, so that means I have little b, right? But obviously, which means there's one chromosome, that I got from my mom, one chromosome that I got from my dad, each of which has a B chromosome on it. But in meiosis, they split, right? That's what segregation means. Independent assortment means when my chromosomes line up, all 23 pair of them that I'm not going to draw right now, when they line up in metaphase 1, it's not like all the mom chromosomes are on one side and all the dad chromosomes on the other, right? Independent assortment means not that. That means this one might be mom, this one might be dad, and maybe this one's also mom and this one's dad, but maybe this one's dad and this one's mom, right? They all line up independent of each other. So we're all caught up. So basically everything on the slide was a review. But we're trying to, again, bring this all these conversations together here at the end of the chapter. So any questions about this review I just gave you on this slide?
No.
All right, so here's kind of a picture of what we're saying. Again, we've already talked about a dihybrid cross where we looked at peas, right? We looked at the peas and we looked at the color. Are they yellow or are they green? And then we looked at the shape. Are they smooth or are they wrinkled? And what we learned when we talked about independent assortment is those two genes that control these things are on completely different chromosomes. Therefore, they go independently of each other, meaning, you could get a yellow, well, I don't have time. We've already talked about it. What I'm saying here, as you already know, is it's not all the mom chromosomes lined up on one side and the dad chromosomes lined up on the other. They go every which way. There is no rhyme or reason. That's all we're saying. And I've already said that. So again, this is just a review. And really, I don't like this picture. It doesn't give enough information. It's too simple. It's so simple that it leaves out too many questions. But we've already saw a more complicated version of this picture way back earlier in the chapter. So I'm just going to skip forward. Oh, there it is. Yeah. So again, what we're saying here, and as you can see, when this thing right here goes through meiosis, right, it's a heterozygote for both of those genes, right? It has a big allele and a little allele, dominant and recessive allele for the R and a dominant and recessive allele for the Y. So no matter what, when this thing goes through meiosis, 100% of its offspring, of its gametes are going to be, they're going to have all the options, so to speak. I don't want to get into it too much and restart the conversation because that'd be a long conversation. So if we just look at the end here, what we're saying here, all right, so this one's, we have two of them, two of this thing's gametes, two of these things gametes would be dominant for the R and dominant for the Y. Two of these things gametes would be recessive for the R and recessive for the Y. Two of these things gametes would be recessive for the R and dominant for the Y. And two of these things gametes would be dominant for the R and recessive for the Y. That's a lot of information. I'm hoping this doesn't overwhelm you. All you really need to get from this picture is the fact that with independent assortment, it's the chromosomes that are doing these things. So the reason why, when you do these Punnett squares and see that you have all these 4 different options, the reason is because these things are on chromosomes, and it's the chromosomes that are going through segregation and independent assortment. And then that, again, same picture, just more information. So I'm going to skip forward. It's everything you already know, except, again, like I said, it's not just alleles segregating and alleles coming back together. Those alleles are on chromosomes. So it's all because of the chromosomes that everything you've learned in this chapter is the way it is. Whew. Speaking of which, this brings us to an exception to the rule. And that exception to the rule is called linked genes. And someone way back at the very beginning of chapter 1 asked questions about this. I think it was Cordone, but I can't remember for sure. But here we go. We're going to jump into it because somebody was asking about the ratio. Because when I said, when we talk about a dihybrid cross, you need to recognize the 9 to 3, hate this thing, to 3 to 3 to 1 ratio. You should recognize that as a dihybrid cross. Right? And then someone asked, well, is it always going to be 9, 3, 3, 1? I said, well, that's a great question, and we'll come back to it. But I did say, as a throwback to remind you, anytime it has 4 numbers in it, then yes, that is a dihybrid cross, as opposed to a monohybrid cross, which would have a 3 to 1 phenotypic ratio. Right? That would just be a monohybrid cross. Anyway, going back to this. Normally, you would expect a 9 to 3 to 3 to 1 ratio with a dihybrid cross, but there's exceptions to that, and those are linked genes. Now, before I get into this, let me say this. Your study guide has some pretty detailed questions about linked genes, and it requires you to figure some stuff out that I figure this is a 100 level class for non-biology majors, so I don't require you to do it. It's in the study guide, and if you would actually like to do it correctly and you need some help, and the information that the book gives you is not enough for you, come and let me know, and I'll help you. But just know that this won't be on the exam. I'll talk a little bit about linked genes, and I'll go ahead and tell you right now what the question might be about linked genes on the exam. It'll say something like, hey, you're supposed to see a 9 to 3 to 3 to 1 ratio when you mix these things. But instead, you got a, 5 to 1 to 1 to 7 ratio, or whatever the numbers may be. Why is that? And the answer is linked genes. Anytime you don't get the ratio you expected, I shouldn't say anytime. As far as we're concerned in this conversation, anytime you don't get the ratio you expected, that 9 to 3 to 3 to 1 ratio, then it is linked genes. Now that you know what you need to know for the exam, let's actually explain linked genes. Linked genes, okay, this might be a question too. Linked genes are located near each other on the same chromosome. So I don't know if you guys can see. Well, yeah, you can. You guys have both this and that. You've got gene 1 that's real close to the center here. Gene 4, which is at the very tip, and then you have gene 2 and gene 3 really close to each other. Those things are linked. And let me try to give you another example. This is going to be so horrible. But imagine that's one of your chromosomes, specifically the one you got from your mom. And then obviously, because it's a homologous chromosome, you also got, this is so bad, you also received one from your dad, right? And the one on the right is supposed to look just like the one on the left, but obviously it doesn't. So there's the one on the left from your mom. Here you've got the one on the right from your dad. Now what we're saying here is, and I'll try to stick with an example I've used before. If your mom has an allele for, big B for bad breath and big H for a hairy back, your mom's got bad breath and a hairy back. And let's say on those same genes, gene 2, good grief, that line's killing me. Those lines are not supposed to be there. For gene 2 and gene 3, again, it's the same gene, but your dad, he gave you the chromosome that had the allele little b, right? So for good breath, and little h for a not hairy back. Now, as you know from independent assortment, if those things were on different chromosomes, they wouldn't be inherited together. But they are on the same chromosome, meaning if you got this chromosome from your mom, right? If you have a bad breath gene, that means in this case, you would also have the hairy back gene because your mom, allele, right? Because your mom has the bad breath allele and the hairy back allele. So you can see why, you're either getting this chromosome on the left or you're getting this chromosome on the right. Does that make sense? Because we know in meiosis, those things split up. And then in fertilization, that's where we get this, right? You got to have a chromosome from your mom. You got a chromosome from your dad. So again, because these two genes are on the same chromosome right next to each other, if your mom has bad breath and a hairy back, and you got that chromosome from her, you're going to have it, right? That's what you're going to have. Now, here's where it gets a little bit trickier. I want to make sure what I just told you sinks in because the trickier part is even less important when it comes to the exam, but you need to know it. This is why genes are linked, right? Because if they're on the same chromosome, then again, if you get your mom's chromosome, then you're going to have every allele that she has on that chromosome. So in that case, they would be linked. Now here's where it gets tricky. Remember, in prophase of meiosis 1, these chromosomes are nice and close, just like they are right now. And they swippy swap, right? They're touching each other, and eventually they swap. So remember, you might have a mom chromosome with a dad chunk on it, and a dad chromosome with a mom chunk on it. Which means, let me do this really quickly so you can try to picture it. All right. Good enough, I guess. Try to do this. Here we go. All right. So imagine, oh, shoot, I shouldn't have erased that part. Let me try to redo this chromosome. Sorry, guys. There's the dad chromosome. All right. That's so horrible. All right. Let's actually use this line this time. Imagine these things are right next to each other. It's prophase 1 of meiosis, and this is the spot where they swap chunks of chromosomes. So again, here's your mom chromosome, here's your dad chromosome, but because these lines are going to kill me. Anyway, in prophase, they swap. So now let's say this mom chromosome right here, from here up, that's still mom chromosome, but then all this, that's the dad section, right? Because they swapped, right? That swapped back and forth. And therefore, this dad chromosome from here up is all dad chromosome, but here you've got mom chromosome. So in this case, if that happens, you will have mom's, right? This chromosome will have mom's gene 1, but then you'll still end up with dad's gene 2, gene 3, and gene 4. That's all we mean by linked. So the very easy thing to think about when it comes to linked genes is that they're all in the same chromosome. But then we get a little bit, one step more complicated and realize not only is it important that they're on the same chromosome, but how close together are they on the same chromosome, right? Because the closer they are, the less likely they are to be separated in the case of crossing over. That's all we're saying. But again, for the exam, before I move on, to remind you, the exam is really simple. Basically, when you get a ratio other than the one you expected, then what causes it? Linked genes. And the other question on the exam might be, where are linked genes located? And they're just located very close to each other. Any questions about that? All right, again, before I move forward, your book's really, I think your book goes into detail about it, because what you can do is you can look at those ratios and see how different they are. And if you notice, perhaps, like you don't see this in the question, you don't see how close they are together, and you have to figure it out from the ratio. So if you have two genes, they're almost always together. And then you have, I don't know, you can tell by looking at the numbers that gene 2 and gene 3 are really close together. Not this picture, but you can tell by your ratio numbers that gene 2 and gene 3 are really close together. And you can also tell that gene 2 and gene 3 are kind of close to number 4, because there's also, you can tell that there's linkage there. And then there'll be some other gene that's on some other chromosome. You're like, well, those things clearly are not linked at all. But I don't know. We don't get into it. I didn't even get into it until I had cell biology in my senior year of undergrad. And it really is, it can be complicated, but it's kind of fun. You can map genes based on their inheritance pattern. Anyway, that's linked genes. Let's talk about sex determination in humans, which we already have. So again, this should be a little bit of a review as well. We're just saying it again in the context of the chromosomal basis of inheritance. Sex determination in humans. Can someone tell me, somebody who has normal sex chromosomes, if it's a male human, what would their chromosomes be? What two chromosomes would they have if they had the normal sex chromosomes?
X and Y.
Perfect. And if they're a female and had normal sex chromosomes, what chromosomes would they have?
XX.
Perfect. And there will be questions about that on the exam, so make sure you know that. But yeah. So again, this is a repeat of what we've already said. I'm going to go through it quickly. Many animals, including mammals, have a pair of sex chromosomes designated X and Y that determine the sex. For humans, males have one X chromosome, one Y. Females have two X chromosomes. Human and male females both have 44 autosomes, which are the sex, all the chromosomes other than the sex chromosomes. Nothing on this slide is new information, so I'm going to skip past it. I will say one more time, just like I did last time, I showed you a slide similar to this. This is very fascinating stuff, in my opinion, if you want to look it up for independent work. For humans, having an X and a Y makes you a male. For other animals, if you were to look like insects, for example, they have completely different situations. Sometimes it's all about the presence of the Y chromosome. It doesn't even matter. You don't need the X and the Y. It's long and complicated, but if you're interested in it, look it up for independent work. There you go. There's the X and the Y chromosome. The Y on the left, the X on the right. This is for humans. So again, now that we've had that brief reminder of what sex chromosomes are in humans, let's talk about sex-linked genes. This makes things even more complicated. Well, not the definition. The definition is pretty simple. What is a sex-linked gene? It is a gene located on a sex chromosome. And that'll be your next word for attendance, sex. A gene located on a sex chromosome is a sex-linked gene. Most sex-linked genes are found on the X chromosome, not the Y chromosome. Can someone look at this picture and take a wild guess why most sex-linked genes are found on this chromosome and not this chromosome? But don't overthink it, because if you don't overthink it, it's easy. If you overthink it, I guess it could be difficult. But just look at the picture. Can someone guess why most of them are on that one on the left and not the one on the right? Don't be afraid to guess. Surely somebody is looking at this and thinks they know it, and maybe they're just afraid to say it.
It's bigger.
Yeah, it's that simple, right? It's bigger. All the genes are written in chromosomes on DNA, right? And that's DNA and protein, right? The bigger it is, generally speaking, the more genes it's going to have. So yes, it's just that simple. Not that I'm going to ask you that, but I was just hoping somebody could, use a little bit of common sense and figure some stuff out. Yes, it is bigger. So we have more genes on the X chromosome than the Y. Here we go. Let's get into some specifics here. Some human conditions, including red-green color blindness and hemophilia, and a type of muscular dystrophy, they result from sex-linked recessive alleles. I haven't done this in a while, but let me do it. I'm going to put an X through this, just because if you're taking notes or watching the video, that'll help visually remind you that I'm not going to ask you that on the exam. I'm not going to say, hey, give me an example of a sex-linked or of a disorder that comes from a sex-linked gene. I'm not going to do that. But we are going to talk about them, just so you know. The first one we're going to talk about is red-green color blindness. This is a common sex-linked disorder caused by a malfunction of light-sensitive cells in the eyes. Again, I'm not going to ask you any of that, but now you know that's what causes it. Usually it's the male that's affected, and I'm going to explain why. Let's just put an X through this whole thing. Heterozygous females have some defects too, but for the most part, we're not even certainly don't say what number it is. But when you're looking at this, anybody who cannot see what number that is. All right, so none of you are red-green colorblind. Anybody want to tell me what number that is?
7.
Yeah, good. Yeah, it's a 7. So you are not red-green colorblind. Yeah, so let's talk about it. Because they're located on sex chromosomes, sex-linked genes exhibit unusual inheritance patterns, right? Because now we're talking about not only do you get the allele or not get the allele, now you're either getting an X chromosome or a Y chromosome as well, right? That's the reason you either got the allele or you didn't. So because of that, you'll see some patterns like, oh, usually males have this or usually females have that. And we're about to talk about these patterns. So here we go. We're still talking about color blindness, and we're looking at this mating. Oh, man, I'll go through this quickly. Here we have a normal female, right? Hence the big N, big N. She's got the dominant alleles, 2 of them. She's homozygous dominant. Here we have an affected male. This dude's colorblind. Now, notice there's only one N because there's only one X chromosome for him. He's got the little N, the recessive. Now, when they mate, this male, again, those alleles are going to segregate in meiosis. So half of his sperm are going to have the X chromosome, which happens to have the little N. Half of his chromosome is going to have the Y chromosome. Obviously, that has nothing to do with the N. Now, when this female goes through meiosis and she makes her eggs, yeah, technically those are going to split, but it doesn't matter because no matter what, all she has to offer is that big X, the X chromosome with a dominant N, right? So no matter what, all of her eggs, 100%, are going to be with a big dominant N chromosome. Then we look at the possible combinations of fertilization, and we have big N, little N coming together. It's all right, that's a heterozygous. female. We have a big N and a Y coming together. So there we have a normal male. Here we have a big N, little n coming together again. So again, we have a heterozygous female. And then again, we have the big N and a Y coming together, so an unaffected male. So you might get a question like this. All the information you need will be given to you, and then you'll just have to do a Punnett square. And the question might be, what are the chances of this couple, this particular couple, having a colorblind female? And the answer is 0. Because this is recessive, a female would have to be homozygous recessive, little n, right? And there's no way you can have 2 X's with little n's. But if the question said, what are the chances that you'll have a female who's heterozygous? That's a 50% chance, right? 50% chance is going to be this, 50% chance is going to be that. What are the chances that you'll have a colorblind male? 0%, right? Because the only way you can have a male is if this male inherits that normal X chromosome from the mom, which means for a male, all you have is the one N allele, therefore it's normal, right? So I wish I had more time to talk about it and give you some more examples. But yes, you might have 1 or 2 questions like that on the exam where I give you all the pertinent information. So you don't need to memorize color blindness. I'll give you the information you need to know. But then I'll say, what happens when this person mates with this person? What are the chances of them having an offspring with this or that? And there's another example. Again, this time the female is heterozygous instead of homozygous dominant. And this time the male is normal as opposed to colorblind. And then you can just look at the Punnett square and see the chances of this, that, and the other thing. And then yet another example. Here we have a colorblind male mating with a heterozygous female. And again, I'm not going to go through it, but I recommend you look at the PowerPoints on Google Classroom and you go through it. Maybe even meet me during office hours and I can explain it for you if you want. And that will bring us to the last word for attendance today, which is going to be history. And that's it. I will be online for office hours today as usual. I'll also be in my office 9 to 10:40. Please get your lab done. Otherwise, if you don't, you need to be online at 2 p.m. If you are late, you're going to get half credit just for showing up late online. If you don't show up, then you're only going to get half or 1/4 credit for your lab. So you might as well either do it or show up. So please don't lose points just because you couldn't show up at 2 p.m. For a very quick lab. So any questions? All right. Hope you guys have a great day, and I'll either see you at lab or see you on Friday.
Have a good one.
All right. Thanks. You too.